How do we avoid pathogens? When we look at the disease triangle, we have to have a virulent pathogen. In order to try and not have disease, what we can do is plant in a manner that we avoid pathogens. You can either plant in an area where the pathogen doesn't exist, or you can figure out another mechanism by which we do it. These are often called cultural control methods. You can avoid a pathogen such as cedar apple rust if you've got apple trees by making sure you don't have any cedar in a certain area. That is a cultural control mechanism. All you have to do is control the way things are growing. Planting date can make a difference. A change in a planting date by a week or two might make a big difference in avoiding a pathogen. We're looking at a cultural mechanism by which we can avoid things. Pathogens can sometimes be avoided by choice of planting site or the timing of planting. Things like soil pH. Soil pH makes a difference in nutrient availability. If the pH is better for nutrient availability, you can avoid the pathogen. If you've got a pathogen that's in the soil, you can avoid the pathogen by possibly modifying the pH. Fertility levels. We always talk about nitrogen and how you've got to be very careful about nitrogen levels. If you get too much succulents, that favors the pathogen. Keep your fertility levels in the right area, and that can help you avoid pathogens. Pathogen avoidance. Very important in the way that we deal with disease. Another part is how do we exclude pathogens? In pathogen exclusion, we're trying to keep them out of an area. There are legal methods. A legal method means you pass a law saying you can't bring it in. Once you pass the law that says you can't bring it in, then you just have to enforce the law. Every time you go out of the country and you come back in, they ask you if you've been on a farm. If you say yes, they want to see your shoes. They're going to spray your shoes to make sure that they kill off whatever pathogens you might be bringing in. One of the legal methods is what we call a quarantine. If you want to bring plants or animals into the United States, some of them have to have a phytosanitary certificate with it. Some of them have to be quarantined. By quarantine, what that means is they take it and they keep it in a specific area for about 90 days to 180 days to make sure that there are no problems with it. We have quarantines, which are a type of legal method. Quarantines basically are laws which are passed to control the entry of pests into an area or to provide for the elimination of pests in a given area. We exclude pathogens. Citrus canker is a good example. Citrus canker occurs in various places in the world. Wherever citrus canker occurs, you cannot export fresh fruit out of that area going to another place that grows citrus. They've got laws saying you can't do that. You can import orange juice. That's not fresh fruit. You cannot send fresh fruit from Florida to California. It's against the law because they grow citrus in California and they don't want any diseases that might occur in Florida. We have laws and regulations on this. When you're driving down the interstate, you often see plant inspection stations. They're looking at plant materials, trying to inspect it and make sure that there's no problems with them. South American leaf blight in Indonesia is another. Heavier rubber tree originated in South America. They took the seeds from it to the Kew Botanical Gardens in England. From there, they took them to Indonesia, where they planted them. The viability of the seeds wasn't great. They did get a few to grow. From that, they've been able to create a tremendous industry in Indonesia. In Indonesia, they do not have a pathogen that occurs in South America. In Indonesia, production rates are about 10 times that what you get in South America, mostly because of the pathogen. If you want to bring the pathogen into Indonesia, they have got a law saying that if you get caught, they will put the individual to death. It's that serious a problem. When you look at this, you've got to be very careful as to what they will let in, what they won't let in. You want to take things to Hawaii, they don't want them because Hawaii's got a very interesting culture. They don't want things coming in and taking over. If you fly into New York City, a lot of times they don't care what you're bringing in because in New York City, as soon as the cold weather hits, whatever it is you brought in from a tropical area is going to die off. We've got laws that allow for things to be permitted in certain areas and things not to be permitted in certain areas. These are very important as far as excluding pathogens from an area. How do we exclude pathogens? One of the things we deal with is pathogen-free seed. You have to make sure that the seed is free of pathogen when you bring it in to make sure that you don't bring in that particular pathogen. Over here, we've got soybean seed. On the right-hand side, it is infected with Cercospora kikuchii. On the left-hand side, it's good seed. Sometimes to try and decrease the amount of pathogen you might be bringing on it, they make you do a treatment on it to try and kill off whatever's in there. This shows you seed that has pathogen associated with, and this has been placed on agar. You can see that the pathogen is growing. This is bunt of wheat. You can see on the right-hand one how it's got this material growing on the inside that is stained dark. That is pathogen. So I want to make sure that you don't have that particular type of material. How do we exclude pathogens? Pathogen-free vegetative propagations. These have to be certified. A lot of times we will grow things in culture media. If you have material that has pathogen in it and it's something like a viral pathogen, one of the ways you can get rid of it is you can take the meristem out of that area before the virus gets into it, grow it on culture media, and then the plants you get out of it are now free of that particular pathogen. We've got mechanisms in here that we can use. This shows you another type of culture media. This is material that originally started out from cells. By modifying the hormones, you can now grow the material without any problems. This is a little plant in a tube. This is produced so that you can have pathogen-free vegetative propagation material. There are cultural practices that we use to exclude pathogen. One is called crop rotation. Crop rotation is where you start out with an area. You're going to plant something like soybean, then next you're going to plant corn, next you're going to plant alfalfa, then you may go back to soybean. When you do this, you change the constitution of the soil and the plants that are in there. That suppresses pathogens. Crop rotation is often used as a cultural practice to reduce pathogen. We have fallowing. Fallowing is where you don't plant anything. If you don't plant anything, nothing's going to stimulate the pathogen to grow, and then you can reduce the amount of pathogen you have. We have flooding. I did an experiment once where it got flooded out. I was trying to monitor the growth of a fairy ring in the soil. The area got flooded for 45 days. The 45 days created anaerobic conditions. The fairy ring was gone forever. Sometimes they will flood things out like rice paddies. That often kills off many things that are aerobic. This is a cultural practice. No chemicals involved in it. We've got host eradication. We talked about this with stem rust of wheat. They eradicated barberry. Taking the barberry out, it stops modification of the pathogen. It's no longer able to do problems that you had. We've got volunteer plants. Those would be things like a corn plant in a soybean field. The corn plant was planted last year. You didn't get all of the seed out. A couple of seeds fell off. All of a sudden, you've got a volunteer plant in there. Sometimes we do what is called roguing, which means you walk around the field and you pull out plants that are problematic. Then we've also got the alternate host. The alternate host is another plant on which that particular pathogen can survive. Many times these might be weeds, but it can still grow in an area. By getting rid of those particular hosts, you can keep the material out so that you keep it from continuing on that cycle. By doing that, you're going to decrease the amount of pathogen you've got. Plowing is another cultural practice we do. In plowing, you bury the material. It is going to cause it to decompose. As it decomposes, you have other microorganisms that come in on top. They help suppress whatever pathogen you've got. Now it's got competition. Many times the pathogen is not able to survive the competition. We have sanitation. This is where you clean up. When I was a kid growing up, we'd rake up the leaves in the yard and we'd set them on fire. They don't like you to do that anymore. Actually was a form of sanitation as it destroyed pathogen. By the same token, you can take that material, you can clean it up, you can turn it into a compost heap. That is a good methodology to reduce pathogen. This shows you a field where they are plowing it and they're paring it. It's also flooded out. The flooding out is going to decrease pathogen because you create anaerobic conditions that are not favorable for the pathogens. This is a gentleman taking his logging truck. He's washing all of the dirt off of it to make sure that that material is not transmitted from one area to the next. This is a cultural sort of thing because basically you're not using any chemicals. You're just using a high pressure washing. And as you knock all of the soil off, it will take pathogen away.